Hello everyone, welcome to Bioinformatics Practicals. In this session, we will be using the ORF finding tools for finding the open reading frames. This practical is given in the 11th item that is provided in the list of practicals. So for analyzing the ORF of a given sequence, you need to go to the ORF finder in the NCBI database. Then there you have to give an input DNA sequence for ORF finding. So this is the ORF finder reading frame. Usually you I'm finding an open reading frame, you need to give a larger input sequence, not a single gene sequence. So usually a segment of the DNA with a start and end needs to be given as an input. In this case, you can see that previously we downloaded one a segment of the chromosome in one of the previous practicals. So you can browse any gene in the NCBI database, and that gene segment can be downloaded. Here in the previous segment that we downloaded for GenMark analysis, this is the chromosome ID, the chromosome ID, and this is the start and end of the gene, the next numbers. So from this title, NC and the code, gene code and the chromosome code can be given here. So this is the accession number of the chromosome. Now the remaining, that is this portion is the gene number. So that is from to. So for every chromosome or for every region of the DNA that you want to analyze, especially if it is in a chromosome, larger chromosome, you give the chromosome accession number and start and end of the region of the sequence. So this can be obtained as explained in one of the previous practicals. So if you give this information, the accession number and the start and end from which region of the chromosome to which region, then submit. It found 13 ORFs, open reading frames. As you know, open reading frames were a part of the 6 reading frames where we have 2 complementary strands of the DNA. Each strand can be read in triplets in 3 ways. That is, the base 1, 2, 3 can be a single codon, 4, 5, 6 can be another codon, or it can start reading from the second base, so that 2, 3, 4 will constitute first codon, and 5, 6, 7 will constitute the second codon. Similarly, so every DNA strand can be read in 3 fashions if you are reading as triplets. So that constitutes 3 reading frames possibilities in a single strand. So the complementary strand is also having another 3 reading possibilities, and that is open reading frame. You know the concept. So here, if you are clicking on display 6 reading frame, add 6 reading frame track. So these are the 6 reading frames. On the plus strand we have 3 reading frames. On the minus strand, there also we have 3 reading frames. And if you are reading this strand, these are the different proteins that is coded from that region. And the other regions it will encounter termination codon, so that will not be coded for. So these genes or these are the fragments that is coded. This region of the gene is coding for some proteins. So that is the open reading frames. So this is the largest one, ORF2 of the plus strand. That is the largest reading frame, ORF2. The next one is ORF12. That is another one. It codes for the nucleotide length of the ORF2 is 600, which codes for 199 amino acids. ORF12 codes for 199 amino acids, and the length of the ORF12 is 597. If you click on this fragments, it will select that region of the DNA, and this is the sequence of the peptide that is coded for. Okay, so this is how you can find the open reading frames, and the corresponding peptide sequence is actually displayed here. And if you are interested in finding more information, you can click on a smart blast from here or another blast. It will blast it taking the Uniprot KB or some other databases. And if such a sequence is there, it will give you the information about that sequence. Okay, this is the result. You can see the smart blast result where our unknown sequence is aligned using a smart blast with some particular organism, the gene of the organism, and it is a hypothetical protein. It is not actually a protein which is translated and or it's identified. It is a hypothetical protein, and that sequence is aligned, and a blast is performed using that ORF. So any protein or any region of the DNA, any fragment of the DNA can be computationally translated, and after finding the open reading frames, and possible protein sequences can be translated, and even that translated region can be blasted in the database for finding similarities with the sequences existing in the database. And in this case, it is not, it's only a hypothetical protein. So actually that protein doesn't exist in the database. That's why we don't have much hits in the database. So any DNA sequence, any region of the chromosome can be given as an input for analysis in open reading frame finding, and such reading frames will be displayed in this graphical representation. And by selecting that concerned reading frame here, the reading frame will be selected here, and also the protein sequence will be displayed here. And this is only a hypothetical protein sequence, and actually that protein may not be expressed. So this is about ORF finding using NCBI ORF finder.